This is my presentation on flow cytometry and the characteristics of it. First, we have an overview. It is a technology that provides rapid multiparametric analysis of single cells in a solution. It utilizes lasers as light sources to produce scattered and fluorescent light signals that are read by detectors like photodiodes or photomultiplier tubes. And these signals are then converted into electronic signals that are analyzed by a computer and written to a standardized format and put into a data recording system and shown as a scatter plot. Cell populations can be analyzed and or purified based on their fluorescent or light scattering characteristics, and a variety of different fluorescent reagents are utilized in flow cytometry, such as fluorescently conjugated antibodies, DNA binding dyes, viability dyes, ion indicator dyes, and fluorescent expression proteins. And over here we just have a little diagram of a basic flow cytometry model. So you have your sample that goes in, and then the sheath fluid is added separately, and then a laser is shined through the sample tube, and then you can see the fluorescence from the stain cells bouncing off of the sample and the scattered light from all the cells that are detected in the sample. Just a little bit more of an overview. Flow cytometry rapidly analyzes single cells or particles as they flow past single or multiple lasers while suspended in a buffered salt-based solution. Each particle is then analyzed for visible light scatter in one or more multiple fluorescence parameters. The visible light scatter is measured in two directions: a forward direction, which is called a forward scatter, or FSC, which indicates the relative size of the cell, and they can also be scattered. The measured can be at 90 degrees, which is a side scatter, or SSC, and this indicates the internal complexity or granularity of the cell. And the light scatter depends on the fluorescence. And over here is just another visual aid, just shows a cell and how they can detect antibodies or antigens on the cell, and that can be used in flow cytometry as well. Traditional flow cytometers consist of three systems that have fluidics, optics, and electronics. Fluidics consist of a sheath fluid, usually a buffered saline solution, which is pressurized to deliver and focus the sample to the laser interceptive interrogation point where the sample will be analyzed. The next is optical, which consists of excitation points or lasers and collection optics that generate the visible and fluorescent light signals used to analyze the sample. So this is how you actually see the light that is detecting the particles within this sample. And then a series of dichroic filters steer the fluorescent light to a specific detector, and bandpass filters determine the wavelengths of light that are read so that each individual fluorochrom can be detected and measured. The dichroic filters are filters that pass light through that is either shorter or longer in wavelength and reflect the remaining light at an angle. So that's how they measure different wavelengths of the sample that is given. Applications in immunology: the way that flow cytology is used is that it is effective for the study of the immune system and its responses to infectious diseases and cancer. It allows for simultaneous characterization of mixed populations of cells from blood and bone marrow, as well as solid tissues that can be dissociated into single cells, which include lymph nodes, spleen, mucosal tissues, solid tumors, and some others. And in addition to the analysis of cell populations, a major application of flow cytometry is sorting cells for future analysis, and it can also be utilized in immunophenotyping, cell cycle analysis, and is able to distinguish cell apoptosis, and it can also be used in proliferation assays. For data analysis, traditionally a two-parameter histogram or a dot plot called gating and analysis is what is used currently, but there is another option that the increase in the number of parameters and complexity in experiments is leading to the use of newer cluster data and analysis algorithms such as PCA, SPADE, and TSNE. These new methods allow for useful information to be extracted from the high-dimensional data that is available from flow cytometry. And over here we just have a scatter plot that indicates whether a sample is going to be correct or not. So in the first one, you can see that the FSC is too low, so it kind of skews the scatter plot to the side more. And in the next one, the FSC is too high, so there's not much data here as compared to the first one. And the last one you can see is where the FSC is actually correct. So we kind of have a even distribution here, more so than the others. The SCS format was developed in 1984 to standardize flow cytometry list mode data files. This conventional flow cytometry analysis consists of drawing a region around a population of cells or gating and applying that region to other parameters within the experiment. This allows specific groups of cells to be selected for further analysis of other markers. For the scatter plot analysis, with a forward scatter, the light that scatters past the cell will denote the diameter and volume of the cell, and a side scatter will determine and inform about the granularity or complexity of the nucleic structure within the cell. And then this last slide just shows the sources that I used to get this information. Thank you.